Good morning, everyone. We'll get started in a few minutes, just allowing people to join. Okay, I think we'll go ahead and get started. Welcome everyone to the fourth webinar in our Rapid Formulation Development series. If you haven't seen the other ones or you want to see them again, they are available, both the videos and the slides, on our website. So you can go to the website and look for those. Just a little bit about FreeThink Technologies. We are the leading laboratory for solving complex stability and formulation challenges using world-class science to speed products to market. Our mission is to use world-class science to speed better products to market, and that includes using our ASAP Prime stability software and packaging, shelf life determinations, as well as ADAP, which is our formulation development. ADAP is accelerated drug advancement process. It's rapid and material-sparing formulation design. There are 4 aspects to that: target product profile, solubilization development, stable formulation development, and the control release development. And so if you want to learn more about us or reach out to us, here we are on LinkedIn. There's our phone number and our web address. Our speaker today for the oral controlled release formulation is Dr. Sherry Shamblin. She received her BA in chemistry from the Great College of Worcester and her PhD in pharmaceutical sciences from the University of Wisconsin-Madison, and conducted postdoctoral research at Purdue University. She worked over 25 years at Pfizer and has extensive experience as a scientific leader in oral drug product development. Dr. Shamblin is a recognized expert in the development of oral controlled release formulations and the use of enabling strategies to increase API solubility and in vivo exposure. Throughout her industrial career, she has worked collaboratively with discovery and commercial asset teams to leverage drug delivery technologies to achieve specific clinical objectives and bring new medicines to market. So I'm going to turn this over to Sherry. As she gets all set up, but just remind folks that there is a button at the bottom of your screen that's Q&A. So if you have any questions, feel free to enter those questions while the webinar is going on, and we'll address as many as we can at the end. So I'll turn it over to you, Sherry.
Thank you, Jennifer, and I'd like to thank all of the FreeThink team for the opportunity to join in and be part of this series. Before I get started, I just would like to say a few words. So I have worked in the pharmaceutical industry for many years, and last year I retired from Pfizer and started working as a consultant with my own company, Alurites. We provide premium technical consultant services for both design and development with both immediate release and also control release medicine. I can offer services from a wide range, from early discovery support all the way up through commercial and launch. So if you anything I can help out with, feel free to contact me. I provided the name of the company and my contact email there on the slide. So without further ado, let's get to the more interesting thing, which is to talk about oral control release formulation. Today, you'll learn first, we're going to talk about the drivers for control release. And today's focus is primarily oral, but many of the same principles will apply. Having looked then at what the drivers are, I'll step back and talk about a range of control release technologies with a very kind of focus on the most common types of control release. And then a little bit about how do you choose? Because there are quite a few things to choose from. What's the right technology for your situation, your drug, your stage of development? And then just a quick wrap up with some tools and tips for development and progressing a control release formulation. So first, when do we talk about, what triggers the need for control release? Probably the most common is to reduce the frequency of dosing. Here, I'm showing a graph of what a typical immediate release formulation might provide, a rapid Cmax, hopefully rapid absorption, and typically a decrease in absorption, decrease in plasma concentration throughout time. And if that is not suitable for the type of coverage, there's a need then to take the drug, immediate release drug, 2 or 3 times a day. What controlled release can do is slow down that input rate, reduce the Cmax, and also sustain the concentration. And that can help maintain coverage through the therapeutic window. So that's one of the reasons, is reducing the frequency of dosing. Sometimes there's a need to reduce local irritation in the gut. So blunting Cmax by slowing down the input rate using controlled release can increase the toleration, the safety, and also sometimes even increase efficacy because you're increasing the time above a certain critical threshold, often referred to as Cmin. And a lot of times the goal is to reduce the Cmax, so give a more steady concentration of delivery throughout the time. Sometimes there's a need not due to safety or efficacy, but perhaps the drug is unstable in a gastric environment. So delaying the release of drug past that acidic environment can also be a driver. Finally, one of the more it's increasing is a need to deliver drug to various regions of the GI tract. Sometimes it's colon targeting for different types of therapies that will help with, say, Crohn's disease, that type of thing. Sometimes there's just a need to bypass a certain area where absorption, or get to an area where absorption is expected to be better. And sometimes the colon is a nice target for that. And given all these different needs, there's then a variety of profiles. Sometimes you'll delay an immediate release. Sometimes you're sustaining for a long period of time at a steady rate of delivery. And sometimes there can be need for pulses. And finally, on the right, lower left, there might even be a situation where you'll take a slow controlled release in combination with immediate release to give full coverage over time. So moving on, before we go much further, it's important to think about, if we're going to slow down release via the oral route, the need to deliver drug likely to different regions in the intestine and even into the colon. So one of the first things we need to think about, and here I'm showing a graphic of the GI tract, is that as we transit from the stomach into the lower intestine, a couple of things happen. There's differences in pH. In the small intestine, it tends to be 6.5, and slightly higher in the large intestine and colon. There's a much greater differences in fluid volumes, so much less water is available to dissolve drug. And also there's a change in sort of the GI physiology. So typically we think about the duodenum and the small intestine as an area of absorption, or sometimes referred to as the absorption window. In the large intestine, the junctions between the epithelial cells become much tighter. And the job of the colon is really to keep things out. The primary job of the colon is to pull water out of the food and to release water. So there's a very different environment, as I said, between the amount of water, the extent of absorption that can occur, partly because of surface area, as well as these tight junctions. And there's a lot less fluid movement or mixing in the lower intestine. So this can create some challenges as we think about the need for drug to first dissolve and then to be absorbed. And so thinking about the dose volume, which is defined as the dose, the total dose in milligrams divided by the solubility, we come up with a dose volume. And you'll see soon how that can become a really powerful tool to transition from, okay, I think I need control release, but then how do I know if control release is going to be even feasible? So I really like the layout, the construct from Avi Tambre. He published this in 2005. It breaks it down into some simple things you need to think about in terms of feasibility. So first is solubility for the reasons I just mentioned. So thinking about what's the total dose that's available that we need to deliver, what's the solubility of that dose, and the duration of delivery. How much of that dose could potentially be delivered lower down in the GI tract where there's very little volume and mixing. Secondly, we need to think about the permeability. Sorry, and forgot to skip over here. The pKa and pH of that can be important, and we'll come to that later in the slides. The permeability is clearly important. The more permeable, it's going to help in that region where water levels are low, tight junctions come into play. Efflux is also important. So the potential for drug to be kicked back out into the intestine, and that can change a lot throughout the GI tract. So understanding that and the role of PGP. Stability, we always worry about stability, but there's some additional considerations when we start looking to materials like polymeric materials can tend to have some impurities and some functionalities that may not have been screened in your initial looking at stability. So something to think about there. Enzymatic stability in the lower GI tract can play a role in potential drug degradation. And finally, we need to start paying a little more attention to metabolism. Certainly, the rate of elimination is something that may be triggering the need for controlled release, but we also need to think about the mechanism of elimination, and particularly if it's primarily hepatic, so drug is eliminated through potentially there could be hepatically, there can be first pass metabolism, which could play a role in the extent of bioavailability in a controlled release relative to the immediate release. So these are just some important things to think about. So how does that actually play out into risk? So in the same feasibility assessment, you can start to look at, well, what's the challenge here? I'm not going to go through everything, but we think about doses first, a very low dose. It can be slightly more challenging because of stability, because of potential first pass elimination. Really, the sweet spot is the 10 to 20, 250 milligrams. And above that, it just becomes more challenging because typically there is more functional components in a dosage form. And so tablet burden can become challenging. Also, just typical of most drugs, the solubility and permeability that is designed in can make delivering doses greater than 300 milligrams more challenging. Is it possible? Absolutely. There are commercial drugs that have doses much higher that are delivered with modified or controlled release. Just something to keep an eye on. The dose solubility we already talked about. So again, sweet spot here is one mil, you've got a lot of options. As you start going into the 100 and 1,000 milliliters, how you deliver it, the way you deliver it becomes a little more critical. And above 1,000 milliliters, you're getting into a region that can be quite challenging. Regional permeability, again, sweet spot there defined. And then thinking about the half-life. So a much longer half-life is pretty easy. Likely you just need a short duration potentially to blunt Cmax or to extend it out. As that half-life becomes shorter and shorter, the need for more advanced or more complex ways of delivering control release come into play and can make overall the likelihood for technical success more challenging. A lot of times people will ask, well, I've got one or two red, is that a problem? Nothing's a problem here. The idea here is just to give you a sense of what the challenges would be. If you take your drug and you lay this out and everything is red, you're up for a challenge and something to think about. So this is just a way to give a sense of upfront, how much effort and how hard am I going to need to work at this? What's the critical decision in choosing the dosage form? I want to share this recent publication because I think it really lines up nicely with what Tombre laid out in 2005. It shows a range of modified release dosage forms in this plot, which is the biopharmaceutics classification system for modified release. The publication reference is shown here. And what it's showing is that both the solubility and permeability, the bulk of these drugs are in the class 1 and class 2. And if you compare where those values of both dose solubility and permeability fall, they're very much in the green and yellow regions that were mapped out in that previous table. So this is just a confirmation that something that was mapped out, well over 10, 15 years ago is holding up quite well. Actually, I think it's more like 20 years this day and age. So moving on from then, so having established some of the likelihood for feasibility that the drug candidate, the goals of the formulation are good ones. And again, going back to one of the earlier talks, the TPP, the target product profile, is really a key here in evaluating which of those objectives that you're looking for then plays into what is the right technology. I'm going to just give you kind of an overview first, and we'll dig a little more deeply into each of these. But the most common control release technology on the market is a matrix tablet. It's pretty simple. It relies on widely available polymers to control the release. Multiparticulates is probably the second most commonly used modified release, in part because it's versatility, and there are a lot of different types of multiparticulates that fall into that category. They typically range from 0.2 to 3 millimeters. They can include beads, granules, mini tablets, microspheres. Sometimes the control release is occurring through the multiparticulate itself. It's slowing the release, and sometimes it's a coating, a functional coating on top of that multiparticulate, and sometimes it's both. The other common way to control release is to delay release, typically an enteric coating, an acidic coating that will prevent drug release in the stomach. This can be applied to both tablets and multiparticulates. You can enteric coat an immediate release tablet for a delayed release that's on a very rapid release lower down in the GI tract, or sometimes you're delaying and then providing a slow release. So we'll talk about both examples there. Less common, but quite powerful is the osmotic pump systems. These systems provide a very sustained and controlled release throughout the GI tract. They can provide a very long duration release, so that's sometimes why this technology is provided. The key here is that it's constant, it's zero order. The rate of release does not change for most of the whole entire dose. And then finally, increasingly, we're seeing gastric retentive systems, which allow a drug to stay in the stomach through a swelling, expanding mechanism by a careful choice of polymers. And that can provide it's nice in a sense that it provides sustained delivery, but it's continuing to deliver to the duodenum, that area of the GI tract where absorption is usually the best. So also quite useful. So let's talk, before we go more in depth into these types of control release, let's talk about fixed first law. The one thing that they all have in common is you're controlling the release of drug using diffusion. And fixed first law, here we have the flux of a material as a function of time. It relies on a diffusion coefficient, which is related to viscosity and the size of the diffusing species. Surface area plays a role here. The distance or the thickness through which a drug has to travel through a barrier, the permeability coefficient, which is related to the nature or often hydrophobicity or hydrophilicity of that barrier, and then the concentration difference. So at time zero, all of the drug is in a tablet or a multiparticulate or on one side of a film, and over time that concentration changes as drug diffuses. And so how that translates in the pharmaceutical design of membranes is that, again, we create a membrane or a film. We can create a viscous barrier through the creation of a gel or a matrix. There's also porous diffusion, where we tends to be a more hydrophobic barrier and create pores through that hydrophobic gel or matrix so that drug can diffuse through. It's a slightly different type of diffusion, referred to as porous diffusion. And then simply we can have drug going through a semi-permeable membrane, again, created by typically a hydrophobic barrier with a more porous or more polar component to that barrier. So having that background, let's look at some matrix tablets. Typically, a matrix tablet is a homogeneous blend of drug, a hydrophilic or more hydrophobic polymer. Sometimes there are soluble and insoluble components for manufacturability. And the top layer I'm showing, or the top part of the slide, I'm showing diffusion mechanism. This is probably the preferred way of creating a matrix tablet, where you've got a dry matrix, again, consisting, to make it simple, drug and polymer, that over time, when water comes in, you start to create a gel with a viscous barrier. So back to Fick's first law, we're creating that viscous diffusion barrier for drug. In an ideal world, the drug has sufficient solubility in that matrix so that it can go into solution and then dissolve in a controlled manner through that matrix. Typically, a diffusion matrix will hold together and stay together until all of the drug is released, and eventually over time it will break up. But in an ideal world, again, that diffusion matrix stays together. Another type of matrix that sometimes you may experience occurs through erosional mechanism. A little less ideal. What happens in this case is that you've got a dry matrix of drug and polymer, but now the drug doesn't quite have the solubility needed to dissolve in that matrix. So a gel layer is created, but then the drug doesn't go in solution. So over time, this insoluble drug will erode the strength of that gel, which will tend to break up to form more like granules or hydrated bits of gel. And so what is occurring here is the erosion of the gel is creating the release of the drug. And so this is less ideal because it's a little less predictable, and it can be sensitive to external environments, shear stresses that occur during digestion. So a little less predictable, a little less likely to be to translate. So once you've designed this release, a little less likely to translate in vivo. It's not necessarily a bad thing, but it's something to be aware of. And in reality, what you typically have is a combination of both of these mechanisms happening. And we'll talk about what properties of drug, in addition to solubility, can affect whether diffusion and/or erosion is occurring or predominating in the release profile. There's a different type of matrix that can also be used, which relies on the use of a wax. This is useful for drugs with very high solubility, where sometimes the polymers available aren't adequate to slow down release. In this case, the matrix tablet's designed by choosing a waxy material. There's a range of them. I'm just showing two common ones. I would say the glycerol bohene is probably the most common, where you create a waxy tablet and then rely either on the drug alone or some additional soluble component to create porous channels into this tablet. In these porous channels, water comes in, eventually dissolves the drug, and then drug comes back out through the tablet. The type of release profile is first order, similar to the hydrophilic matrices. But again, this is a nice way to slow down the release of a very soluble drug, especially if a longer duration is needed. So just to summarize some of the attributes of the matrix tablets, probably the most important thing is it's fairly simple. There's a wide range of materials, and that can be helpful both achieving the duration, as well as if you have any type of incompatibility or stability problem, there are other options, other means of achieving that same type of matrix. A matrix does not require a functional coating, but they can be used for additional ways to control the release. Some of the challenges of this technology, as I mentioned, is that the solubility of the drug can play a role. And so if there are changes in solubility as a function of pH, that will affect the rate of release. So the rate can be variable over time. Eroding matrices are more variable because of the potential to have different releases in different, whether it's dosed with food or without food, that digestion can really affect the rate of release. And therefore, predictions of in vivo release from in vitro can be a little more challenging. This typically can be overcome by iterative approaches, but getting it right the first time is a little more challenging with the matrix tablet. I'd like to just spend a moment talking about the challenge with ionizable drugs. And maybe it's not a challenge, but it's a consideration. And this is important because depending on which survey you look at, more than 50 to 60, even as much as 70% of drugs are ionizable or have an ionizable moiety. So likely that means that the solubility of the drug changes as a function of pH. And certainly for many of the drugs, changes over the physiological pH. So here I'm showing a function, a theoretical plot of the solubility as a function of pH. And so if we just let's look at the acidic materials. So if we have an acidic drug with, say, a pK of 4 or 5, in the stomach environment, it's unionized, so solubility is low. But as the drug transitions, or even the dosage form transitions into the intestinal tract, that pH will increase, and now the drug becomes more soluble. So now assuming that this drug is embedded into a polymer, the rate of the polymer, the rate of diffusion through that polymer will be faster in the intestinal environment as compared to the uh, acidic environment. So that may just be one thing that could happen, but you could also have a completely different mechanism. It may be more of an erosion mechanism initially, but over time that may become more of a diffusion mechanism. The opposite is true for a basic drug where the solubility is high in the stomach. So initially, you might have a really well-designed diffusion matrix tablet. The diffusion is being controlled by the viscosity of the polymer and the size of that tablet. But over time, as the dosage form transitions into the lower GI tract, as the solubility drops, that diffusion rate will slow as drug solubility decreases. And so this is something to think about in the terms of ionizable drugs when thinking about design of a matrix tablet. And I thought an example might be helpful here. This is nifedipine, which is a weakly acidic drug. It has a pK of 3.9. So in the stomach, this drug has very poor solubility. And if you look at the in vitro dissolution on the top right, it's showing that even at pH 1.4, the dissolution rate is very, very limited. At higher pH, that dissolution rate becomes very high. It becomes as the drug becomes soluble. How this translates to in vivo is that if you look at the concentration versus time for matrix tablets in the fed and fasted state, the matrix in the fed state, shown in the orange line, has very little release in the first few hours, but over time, that becomes much more rapid rates of dissolution through that tablet with higher pH. That same tablet, when dosed in the fed state, the pH is high enough that in the stomach, in the fed state, that dissolution occurs right away. And so you see much more rapid release. So this has a very positive food effect, this particular formulation. So just something to be aware of. I think it shows a nice example of how different a matrix tablet can perform in vivo with the dosing of food. So that is a good segue into talking about multiparticulates. So what are multiparticulates? I mentioned that these can be a variety of different things. There are wax and polymer matrix-based beads that can be formed through granulation. One of the most common ways to dose multiparticulates modified release is to start with an inert bead to coat the drug onto that, and then to show additional coatings. This is kind of what I have schematically here. You see this inert core, perhaps with a drug layer, and then a range of functional coatings can be applied to control or slow the release. Granules can be made that slow release. These are really helpful in combination therapies and therapies to patients who have difficulty swallowing. All of these types of materials can be dosed coated or uncoated, depending on whether control is imparted by the bead or the granule, or whether there's a need for additional slowing of release, either through a delay with an enteric coating or another type of functional coating that will slow release. One of the benefits of multiparticulates is that not only are there a wide range of achieving slowing the release, but the presentation to the patient can be quite different. So one option is to fill these beads or granules into a capsule. A sachet or stick pack can also be used. Again, this is helpful for higher doses, which may overcome tablet burden, or in patients who have difficulty swallowing. The flexibility that can be achieved, shown here in the bottom of the slide, is that you can have a matrix first order type release. You can have a more controlled zero order release achieved with even an osmotic type of coating on a bead. You can delay the release. So this is really the power of multiparticulates, and usually, not usually, but often the driver for choosing this type of technology. I think I've probably covered a lot of these attributes of them. It really boils down to the versatility and the flexibility that these types of dosage forms offer. One of the challenges is that there are more complex manufacturing processes. There may be more than one type of process involved. So that makes things a little more difficult, expensive than a matrix tablet. Another consideration is that the drug loading might be a little low, so that you have, I guess, you have to think about the potential burden, whether it's a capsule or a tablet or even a sachet, how much needs to be swallowed. So I would say that's not as critical, but something to think about considering the doses. So going back to that feasibility assessment, what is the total dose that has to be delivered by control release? Something to think about here. So multi-particulates have a range of different mechanisms for release. I mentioned earlier the diffusion through pores. Just like wax matrix tablets, wax beads can be designed where you've got a very waxy material which will slow the release of a polar or water-soluble drug. And the way that is achieved is by having a soluble component. Sometimes the drug is soluble enough, but we'll add additional soluble components, sugars, osmogens, to help initiate that water influx into the bead, setting up those water-filled pores, allowing drug to diffuse back out. There's also just diffusion through a polymer. It's a small multi-particulate version of a mini matrix tablet. One of the things to consider in thinking through multi-particulate versus matrix tablet is that size is important. Going back to Fick's first law, surface area to volume can play a role here. So you might imagine that if a really long duration is needed, it may be difficult with a very small matrix bead to achieve a long duration in comparison to a much larger matrix tablet. And that's where coatings can come into play. And that's sort of what I'm showing graphically below here, is that in order to achieve a longer duration, you may need not only a matrix tablet, but the addition of a delayed release or another type of functional coating to slow release out of that. Osmotic pumping can be part of that. Again, you would add an osmogen here, and an osmotic coating can be applied. And we'll talk more about osmotic coatings, but I wanted to mention it here because it can be some approach to slowing down release. And just like in a matrix tablet, you can have eroding multi-particulates, and that's a great design for maybe a shorter duration matrix tablet or matrix multi-particulate. And again, I want to say I'm showing beads here, but the same principles can be applied in some cases to granules and even mini tablets. So how do we make multi-particulates? Part of the versatility is because there are so many ways of making them. One of the most common is the use of fluid bed. This is how you would take an inert bead like a solid sphere, apply a drug layer, and the drug could just be drug onto that bead. It could be drug mixed with a slow polymer designed to slow release. Sometimes it's drug and drug coated followed by a polymer coating that can slow the release, such as an enteric coating. I'm showing a picture here of what these cellet spheres look like. There are other types of inert beads, but the cellet sphere is probably one of the more common ones. In addition to coating beads, fluid bed can also be used to build up or create granules or multi-particulates from different types of combinations of drugs, polymers, osmogens. Another way to make granules or multi-particulate granules is using extrusion, where you're combining the drug and a rate controlling polymer or even a waxy material, extruding those two. So through the process of temperature and solvent and mixing can create a granule. This can also be a way to ultimately feed a mixture of drug, polymer, or drug and wax onto a spinning disc to create a very well-controlled sphere. Or also this can be used for spray congealing. It's like spray drying to create a more spherical, more well-controlled multi-particulate as compared to the granules. So it's just looking at combinations. And this is where the complexity comes into play, is that sometimes you're combining both an extrusion and a spray process. Sometimes you're extruding and coating. Probably one of the simplest processes for making multi-particulates is compression of a blend to create a mini tablet. This is really powerful for a pediatric approach, a kind of a simple way to take, potentially if you've got a matrix tablet, you can take that same formulation and now make mini tablets. It's also a way to create coated mini tablets as well. So those are the processes. Just a quick overview. It's hard to cover everything, but I wanted to show you how those are made. And now I'd like to move on to osmotic tablets. Here I'm showing a kind of a cartoon of an osmotic tablet. An osmotic tablet or an osmotic capsule, those exist as well. And FreeThink is a great great one for this. So if you've got any questions about some of the osmotic capsule capabilities in the early space, I'm not going to go into that here today because I really want to focus more on what's right now available for clinical and commercial use. In all cases, the osmotic tablet relies on the presence of an osmogen, a very soluble component, and commonly sodium chloride is used here. What occurs is that you've got a very soluble component that's going to drive water through a semi-permeable membrane. Semi-permeable membranes are typically formed by taking cellulose acetate in combination with polyethylene glycol, a more polar component. This creates a very strong coating around a tablet. It can be a bilayer tablet shown in the upper left, or in the lower right, a single layer tablet. In the case of the bilayer tablet, this coating protects an active layer, which is drug embedded into a polyethylene oxide matrix. In the lower part of the tablet, in the swelling layer, you've got this osmogen, typically sodium chloride. It can also be sugars, other salts, in combination with a very high molecular weight polyethylene oxide that swells. Initially, going back to, it's always about diffusion. Initially, water diffuses passively through this coating, this barrier, into the core. Once the water sees the sodium chloride or whatever osmogen is present, you create this osmotic pump. So there's now a high concentration of sodium chloride in solution inside the tablet, which continues to drive water into the tablet. Over time, this water starts to cause swelling. And I'm going to just flip to the next slide so that you can see a picture of this as I talk through it. So over time, water starts to dissolve the polymer, dissolves the sodium chloride. That creates a viscous gel or kind of a paste inside the tablet. The viscosity of both the drug layer and the sweller layer begin to drop. So as swelling occurs, there's now this internal hydrostatic pressure as a result of the osmotic force that's driving water in. This hydrostatic pressure starts to move the hydrated, less viscous drug layer up and out of the tablet through a port, typically drilled with a laser, that then releases drug through that and out. Typically, the drug is coming out through a very viscous-like gel. The drug can dissolve in the tablet and be released partly in solution, or if the solubility is not adequate, it can be released as solid drug. Typically, the size of the hole is about roughly a millimeter, anywhere between 0.8 millimeters up to 1.2. So drug can certainly come out in the solid form along with this pasty-like gel. What happens then is that kind of gel that comes out, we sometimes refer to it as a plume, you have a combination of disintegration of that plume and dissolution. And at the very end of this release, you have an intact tablet that has this residual sweller layer. And if you've done things correctly, most of the drug comes out. What's really nice about this type of delivery, this osmotic pump setup, is that you've got this constant rate of delivery, also referred to as a zero order release. Shown on the lower right corner here, you see that there is a bit of a lag in that release. That is the passive diffusion, that is initial, the setting up of the pump. But once that happens, you've got a constant rate of release. Typically, up to 80–90% of the drug is released at the same. And you'll have a little leveling off as that very residual drug kind of diffuses out of the tablet. But this is a highly controlled rate of release. And again, one of the most common reasons for choosing this technology is the desire to sustain at a consistent manner that rate of release. I want to show you what this translates to in terms of a benefit to potentially the patient, and also makes this a little more robust. I'm going back to show you that same nifedipine example. So I showed you earlier that there was a food effect with the matrix tablet shown here, and I apologize, I've lost my laser pointer, so I'm going to just talk through it. I've shown you the data earlier, so you saw the positive food effect with the matrix tablet. When prepared as an osmotic tablet, you see that that food effect has gone away. The profile has changed. It's not the same profile as the matrix tablet, in part because you've got this consistent rate of delivery of the osmotic tablet independent to the food effect. So the drug is being released without any regard to the effects of digestion, both the shear effects, any pH changes. the osmotic tablet continues to release drug at the same rate, independent of all of that. So that's one of the primary benefits. But there's some other benefits to this too, and that it's really what leads up to that is that you've got a very consistent in vitro release that then translates to in vivo. So typically this requires a fewer number of iterations. Now, this ability to achieve a constant rate of delivery in a manner that's sort of independent, both in vitro and in vivo environments, making a very easy translation from in vitro and vivo, comes with a cost. It's very complex. So I mentioned, you saw that if you choose a bilayer tablet, this can mean a bilayer press. So already you're into a more sophisticated way of compressing the tablet. The need for that inert coating around the outside of the tablet that becomes semi-permeable with as it is introduced to water requires solvent coating. So specific types of solvent of coaters that can achieve that. Dealing with the solvent is an issue. And finally, the drilling of the laser hole is a whole different type of process that's not easily or readily available in many manufacturing environments. Now, there are ways around that. I showed an example of a monolithic tablet that can be achieved. You can achieve through a very porous coating and a soluble drug. You can avoid the need for a laser drilled hole, but still, this is a very complex type of manufacturing. The one consideration is that the lag can be a challenge in certain situations. So going back to the target product profile, looking at what your risk assessments are, if that's an issue, that might mean that the osmotic tablet is not the right technology. It's also not a technology that's easily amenable to combinations. There are examples on the market of providing both controlled release and immediate release. So if you've got two different types of drugs that require different types of control release, that can be, or different types of release profiles, that can make an osmotic tablet difficult, a little more challenging. Not out of the question, but something to consider. Might not be the best way to do that. Finally, I want to just touch on a couple of different ways of controlling release. I mentioned delayed release. I just want to spend a little more time talking about how that is done. Most commonly, that's the use of an enteric polymer, which, like many of the polymers, is cellulosic in manner, but has an acidic moiety on it that prevents that polymer from dissolving in the acid environment of the stomach. I'm showing here one type of this. There are many different types of polymers, both in terms of viscosity range, a degree of substitution that can vary exactly when that polymer will start to release drug. So this is just one consideration. There are also erodible polymer coatings and barriers that can be used to slow or delay release. So it doesn't have to be just through pH control. There are ways of looking at essentially achieving it through time by creating a thick barrier. And all of this is about choosing the right type of polymer to give you either delayed based on pH, time, and these can be applied to multiparticulates and also tablets. Finally, I'd like to talk about gastroretentive. These types of systems are being used more and more. The primary benefit of this is for drugs which are expected to have very low solubility in the lower GI tract or poor permeability in the GI tract. They can also be this can also be used when there's concerns about gut metabolism, which I mentioned is one of the considerations for control release. I've shown a nice picture here. The idea is that you design, these tend to start as large tablets, but they're designed so that with hydration, polymers or swelling materials that will create a very large tablet that expands to the point where it exceeds the size of the opening in the pylorus. And so that tablet or that dosage form will sit in the stomach for very long periods of time, allowing the drug, again, still using diffusion, diffuse slowly, then to pass into the lower intestine where it can then be absorbed. And this can allow sustainment for quite a long time. There's a really nice video if you're interested to see how this works. The Graalise is a good commercial example. There's a nice video, and if you're interested in this technology, I encourage you to check it out on YouTube. It gives a great explanation, but also shows the visual. I didn't really want to risk being able to show that here in this webinar today, but I encourage you to look at that. So I'd like to just summarize with a few pointers on how do you choose? So we've laid out, so you've done your control release feasibility. You know a lot about the dose, the properties of the drug. I've shown you there's a wide array of dosage forms available. How do you choose? And so I like to think about it sort of, it's not an easy answer, and it depends. And there's usually more than one answer. It's not like just one dosage form or one technology is going to be the answer. So you really have to go back to the target product profile and think about what's the frequency of dosing, what's the age of the patients, what's the disease state, what other drugs might the patients be taking. Then you have to look at the drug properties. What's the solubility? Is it an ionizable drug? What's the expected permeability? And what do I know about, is this a rapidly cleared drug? What's the volume of distribution? Understanding that and working with your clinical pharmacologist is a really key consideration in thinking this through. And then ultimately, it's also about what's the target release profile? Am I looking to deliver drug to the colon? Do I need to delay release? Is it about blunting a Cmax for toleration? So having all of that in mind is the first step. So almost always, I would say, why not start with the thing with the greatest precedence, if you can? Think about a matrix tablet. It's simple. As I mentioned earlier, there are a wide range of polymers that vary in terms of their chemistry, the viscosity that will be achieved, that gives flexibility potentially for looking at if you have any stability issues, there's alternates. So this is a good place to start. Why would you not go in a matrix tablet? Well, low drug solubility and ionizability could mean that you're going to end up with a food effect. Potentially, if you've got a pediatric or geriatric consideration, if ultimately you've got a requirement to have a pediatric formulation, a matrix tablet may be too large. Now, there is an option potentially to look at matrix mini tablets, so it's not completely off the table, but it's something to consider. High doses. If you've got high dose, high tablet burden in a patient population that's not well suited, where those large tablets wouldn't be acceptable or be able to be swallowed, you might want to think about a multi-particulate that could be dosed as a sachet. Combination products, not out of the question. It's possible that if you've got a matrix tablet with two drugs that can be delivered using the same that require the same rate of release and respond to the same formulation design, it's a possibility. And there are combination products on the market where you've got an immediate release layer and a matrix layer. But when you're starting out with combination therapies, it may be that getting these fixed dose combinations into a matrix tablet may require more iterations, more upfront. So thinking about the flexibility of a multi-particulate may be a better option in that case. So when would you use multi-particulates? Well, we've started to already answer that question, but if your target product profile tells you need this in a pediatric patient or a geriatric patient with a problem swallowing or taking that they're going to end up putting this in food or in a drink, might be something that's worthwhile thinking up front if you have that option. There are also situations where patients of any age have difficulty swallowing. Oncology patients undergoing radiation for treatment of lung cancer can have difficulty swallowing. So perhaps that's a consideration. If you've got a very low solubility drug, perhaps drug or a low dose, putting it onto a bead might be appropriate because that might be an easier way to deal with highly potent low dose drugs. So thinking about that consideration. If you've got a complex profile, if your desire is to target the colon, perhaps a bead that will be delayed lower down into the GI tract could be an approach. Or if you've got multiple release durations. I've had the experience more than once of needing to combine immediate release and control release to achieve the duration. It's very easy to do if you can manufacture an immediate release granule or a bead and then put a delayed or control release coating and combine the two. It gives a lot of flexibility in the early clinical evaluation, and that can translate very easily to a commercial product. Finally, when to go with this osmotic tablet, I've laid out the complexity of how difficult they are to make, but yet there are commercial products because they do have benefits. And it's really likely to be when you've got a really long duration needed. You need to sustain release for a long period of time, either to achieve the right safety profile, or maybe you are targeting some drug very well down in the colon. My rule of thumb is about 8 hours. But below 8 hours, pretty easy to achieve for most drugs with matrix tablets. If it gets longer than that, you might want to think about an osmotic tablet, but it depends on your dose and it depends on solubility. Where another nice place to use the osmotic tablet was when you expect to have a food effect and you can't live with one, that you can't live with a food label on your product. So knowing that you may have a food effect, starting out with an osmotic tablet, it's probably going to reduce the upfront variability and the number of iterations. But again, that complexity will come into play. So it's really these types of things where you're going to have trouble with the osmotic tablet is because the tablets tend to be large. So if you've got a moderate to higher dose, you need to think about whether this will be acceptable in patients that are difficult to swallow or even in very young patients where tablets are not an option. I'm just going to leave you with a few tips here. To think about as you start to go down your formulation. So FreeThink is a great company here to help out with some additional stability compatibility issues. This is really important because in general, the number of materials that are used in controlling release of any technology are much more limited compared to immediate release form excipients. So that if you do highlight a problem with excipient drug compatibility, you may have fewer options. So the sooner that you can identify this, the better off you'll be. I would also say that you need to start the analytical method development early for a number of reasons. One is get those stability flags as early as possible, but also because, and to the third bullet here, extraction potency methods can be more challenging. Use of polymers which gel and swell that combine the drug can make coming up with a good potency method or an extraction approach difficult. So I would say start those early. Using sink conditions early in the design just to be able to understand what the dosage form is doing can be helpful. It may not be representative in terms of what you expect in vivo, but it's a good starting point just to understand what's happening with the dosage form and are you getting the drug out at the rate you expect. As I said, low potency is a challenge, is a problem. So it's something to keep an eye on. And this can happen in part because of the extraction. Drug can remain in the tablets more again because of the viscous barrier that you've created to slow the release. And also because of a loss in drug during processing. I've mentioned that you typically have more complex manufacturing, maybe more steps. And by the time you've blended, maybe tablet, put coatings, it's just something to think about in terms of what to keep an eye out as you're progressing through early stages of development. And finally, just a few other things to consider. The development is iterative. Getting that in vitro, in vivo correlations or relationships initially takes multiple PK studies. So as you're putting together your development plan, make sure that you have multiple PK studies on the plan. When you're planning your clinical studies, work with the clinical pharmacologist. Having an IR, ideal an IV, but at least an IR as a reference is important. There's sometimes a tendency to just maybe put 2 or 3 modified release formulations in early on so that you can establish that relationship between in vitro and vivo. But making sure that you have a good reference point is really critical to understanding what's happening. And then I would say for drugs with moderate to high PK variability, consider the number of subjects and patients. A little more challenging early on when you're doing scoping PK studies, but as you go towards the later pivotal BA and BE studies, starting to put together a plan that will give you more subjects to understand that variability is really critical. And so with that, I just want to highlight a few collaborators that I've worked with over the years here, including Ken. And I want to thank everyone at FreeThink again for the opportunity to talk to you today about control release formulations. And hopefully I've left a few minutes for any questions. So Jennifer, I'll turn it back to you.
Great. Thank you, Sherry. We do have some questions for you. So one is it possible to combine solubilization enhancement with controlled release dosage forms?
It is possible. I would say it's challenging. There aren't a lot of examples commercially where that's been done. Part of the reason that there was that red color in the solubility and the dose solubility is because it is challenging. If you think about the challenge comes from both the ability to, so typically when we improve solubility through something like an ASD or some type of co-solvent system, there's a tendency for that to sustain for a shorter period of time. And so if you're going to take, let's say, an ASD approach, the ability to sustain for a longer period of time is really the primary challenge. There's this tendency for crystallization. The other challenge is that the burden of formulation excipients that's needed. Because now you might be relying on, let's say, a polymer to create an amorphous, stabilized amorphous form, but now you're also adding polymers to sustain release. So it's it's challenging, but I'm not going to say it's impossible.
Okay. Um, and you had mentioned that one way to develop a multi-particulate was to take basically a larger matrix tablet formulation and compress them as mini tablets. So would those give the same release profile?
No, they wouldn't. That's probably, I probably glossed over that a little too quickly. So if you go back to the fixed first law, right? So one of the attributes of the matrix tablets is that they depend not only on the formulation, but the size. So you would expect that as you decrease the size of the matrix tablet to a mini tablet, it will be faster. And so likely you're going to have to maybe change the type and or amount of polymer, but I would say it's a good starting point. What may need to look at is some kind of coating to help with some of that to get that. But it's I probably made that sound a little easier than it actually is. It's a good question.
Okay. Another question is, do gastro-retentive tablets need to be dosed with food?
Um, that's a good question. I do not well, okay, that's going to depend. Because there's different types of gastro-retentive. It was a big sweeping broad category of how they work. Some of them sort of unfold more. Uh, I think my experience, I'm trying to look back to what I did a little survey here. Probably most of them are dosed initially with food, but there was an example of not. So it really kind of depends on the technology that you use, the attributes of that technology, the types of polymers, how quickly they hydrate.
Okay. And then how long can gastro-retentive dosage forms last in terms of duration of release?
Again, that varies. So I've seen what's actually been claimed versus what's been demonstrated. There's examples where it's exceeded 24 hours, but I would say safely precedents would put it more in the 6 to 18 hours. So the idea that it would stay there commercially in humans for a long period of time, 24 hours is probably the actual limit that I would rely on. There are animal products that have been much longer than that, but not in humans.
Um, and then that actually segues nicely into another question. Is there a good animal model to assess controlled release formulations?
That's a great question. I had a slide on this, but it was just too much. So that really varies a lot. It depends on a lot on the mechanism absorption, the type of metabolism, the duration of release. In my own experience, quite honestly, the animal models are difficult. That I've ultimately seen better results and trying to really understand in vitro what's happening in different environments with different degrees of stirring, to understand as best I can the in vitro performance of that, and then using modeling. So using GastroPlus, other types of models to try to understand how that dosage form will likely translate in vivo. I mean, there are situations where it's kind of nice to kind of get a rough measure of it, but it's so different that there's really no one animal model. Ultimately, it's multiple animal models, but generally, my experience is put the work into an in vitro understanding, and that's a better translation, along with some PK modeling.
Um, and then another question, can we expect lower risk of drug-drug interactions with controlled release formulations?
I would, so I'm not sure exactly what that is. Not particularly, I would say. I think that it's actually drug-drug interaction is um, unless there's a specific example that I'm trying to think here, I think you're still going to have those. Now, potentially, if you've, I'm just trying to think of a situation where potentially you're delaying the release of one of them in a manner such that it was, if it's like PGP, let's say, if you're worrying about gut metabolism or efflux, there's a potential there, I suppose, to potentially protect the influence of one for the other. But I'm not aware of if that's been done, but I think it would have to be, I could see that if you're worried about efflux or PGP, gut metabolism, potentially separating, co-dosing, but separating the delivery may be an advantage.
That makes sense. Okay, great. Well, we're out of time. So thank you, everyone. And thank you, Sherry, Maria, Ken, Jessica, for speaking on the series. The slides and video from this will be on our website shortly, and we'll address any questions that weren't addressed during this webinar offline. So thank you all very much.